Bismillahirrahmanirrahim. We start the MCQ topical MCQ questions on the second chapter, which is biological molecules. And I hope this is helpful for all of you. Now let's look at question number 1. Boiling the bones and teeth from dead animals can be used to produce a type of glue. So something very sticky. Glue means something very sticky. The glue is formed from the collagen fibers. Now if you ever had siri pie, and if you sort of after eating it, you see their hands become very sticky if you let the siri pie stick to your fingers. Now basically it is because the collagen, if it was insoluble, now has become soluble. So the helices of the answer is the helices of the collagen molecules unwind. So this is what you have to understand about why it becomes sticky. Then coming to the question 2. What describes the primary structure of a protein? It's not the alpha helix, that's a secondary structure. Dipeptide is only when there are 2 amino acids. Globular structure, no, is the final folding. The specific order of amino acids. Now, I've given you 2 examples here. Now, both of them have 6 amino acids, but look at the sequence is different. One is amino acid number 5, then 10, then 11, then 12, then 14, then 5. This one also has 6 amino acids, but then you can see how their sequence is different. So, it's 2 different proteins. And that is the primary structure of a protein, is the number and sequence of amino acids. Now, question number 3, the diagram shows 4 monosaccharides with the formula C6H12O6. And which diagram shows alpha glucose? Now, you know in alpha glucose, the OH group on carbon 1 is below. So this is alpha glucose if you look at this diagram which I have given you. So alpha glucose. Now this of course I've taken from and I've added another picture to it. This was not in the question. But I mean, you can see it here as well, and you can concentrate it, and you have to see how on 1 the OH is below in alpha, and in this it is above. But 2, 3, 4, 5 all have the same structure. So 2H is above, OH is below, 3H is below, OH above, 4H is above, OH below, and 5 is an H, and then 6 has a CH2OH. So if you look at this question, which one was wrong? I mean, the A was wrong because this was wrong there. C was wrong because this is wrong. So the yellow ones which I've marked is the ones which were wrong and why they were wrong. So this is what you need to understand so you have to know the structure of alpha and beta glucose, not just the one carbon 1, but the rest of it as well. Now coming to question number 4, complete digestion of polysaccharides requires all the glycosidic bonds between the monomers to be broken. Amylase only breaks 1,4, alpha 1,4. So cellulose cannot be because this is beta glucose. That is the first thing you've got to understand. So it had to be only A and B. These were all rubbish. So alpha is only in starch and glycogen. Which row shows how completely amylose? So amylopectin, you know, has 1,4 and 1,6. Amylopectin has 1,4 and 1,6. So it's only breaking the 1,4. So this would be just a little bit of digestion. And amylose only has 1,4. So this would be a complete digestion. Now, if you knew this, you could do this question. If you didn't know it, I'm sorry, you can't do this question. Now, the diagram shows the results for solutions containing biological molecule, which shows contains only starch and protein. So starch is iodine test, protein is biuret test, and that would be D. Veredix test is orange, so that means it contains a reducing sugar as well, but we didn't want any reducing sugar. Question 6, the diagram shows 4 monosaccharides with the formula C6H12O6. Which diagrams do not show glucose molecules? Now, why was the answer D, 3 and 4? And I've just pasted this picture of alpha and beta glucose. You can see that the carbon 2 is wrong, this was wrong, and carbon 3 is wrong. So you can always, you'd have to remember these. You'd have to remember these. Please remember to look at this picture that I posted, and then you can remember it from here, and then you can go back to the question and understand it. So you have to know the structures of alpha and beta glucose. So that is why D was 3 and 4. And let's look at question number 7. Complete digestion of polysaccharides requires all the glycosidic bonds between the monomers to be broken. Amylase breaks only alpha 1,4 glycosidic bonds. Now you know amylose has only 1,4, so that is complete digestion. Most digestion. But amylopectin and glycogen. Now why is this 2 plus and 1 plus? Because you remember glycogen has more 1,6 bonds. So 1,6 will not be broken. Only the 1,4 will be broken. This has more 1,6, but this has less 1,6. So less 1,6 and more 1,6. So that is why the answer is D. Please understand this question. It's not very easy. You cannot be just doing guesswork here. You have to know it. You have to know that amylose, amylopectin, and glycogen. You have to know that amylose has 1,4, amylopectin has 1,4 and 1,6, glycogen has 1,4 and 1,6, and glycogen has more frequent branching. More frequent branching means more 1,6 bonds. Now, question 8, which statements about triglyceride molecules are correct? So you have to look at the incorrect ones. It always contains unsaturated. No, it can be saturated or unsaturated. There's no always. There's no question of always kahani here. Is formed using ester bonds? Yes, that was correct. It does not form hydrogen bonds. Yes, that was correct. Contains the hydrophilic glycerol head that's in a phospholipid molecule, not in a triglyceride. They were asking you about a triglyceride. Triglyceride is a glycerol, which is a 3 carbon, and then it has 3 fatty acid chains attached to it. That's why it's called tri-3, tricycle. Then question number 9. The table shows some features of collagen and hemoglobin. Which row is collagen? It's a fibrous protein made of 3 polypeptides and it's insoluble. So the globulin one was all rubbish. So you immediately cut that out and then you narrow down to A and B. And if you know the structure, then you would know it. Then coming on to question number 10. The ring structure of glucose can also be represented as a linear structure as shown in the diagram. Which of the sugar molecules could be represented by this formula? Well, of course, you've got to count the carbon. So it has 6 carbon. You see, 1, 2, 3, 4, 5, 6, and you know only glucose is a 6 carbon, ribose is a 5 carbon. It's a pentose sugar, and sucrose is a disaccharide. It's made up of glucose and fructose. So it's something C12 because there's C6H12O6, and then fructose is also added to it. So it was one only. Then coming on to question number 11, what is the number of carbon atoms, minimum number of carbon atoms? That's how we read quickly and we read wrongly. So the minimum number of carbon atoms is, you've got to remember a structure, NCC. And then there is something here, and then there's an H here, there's an H here, there's an H here, and there's a C double bond O, and then there's an OH. So it's only these two. Of course, there can be C here as well in the R groups. There can be, but it said minimum number. So when we are assuming that the R group does not have any carbon in it, so this is what we had to understand. Now, if you look at question 12, it says HIV protease is an enzyme produced by the HIV virus. Two identical chains of 99 amino acids form the enzyme. When it says two identical chains, immediately what should come to your mind, if there are two or more chains, then it's a quaternary structure. So this one is rubbish. So you cancel out the one which is totally wrong, and then you reread the question. In each chain, amino acid is 25. So this enzyme is made up of two polypeptide chains, right? So there are two polypeptide chains. And then it says is that, enzyme amino acid is 25. So there's an amino acid here, 25, and amino acid 26 and 27 form part of the active site. Which orders of protein structure control the shape of the active site? Well, because if there were two, then there has to be a primary, secondary, tertiary, and a quaternary structure. So that is why this was the answer. It isn't just, you see, the primary has to make the chain, and then there's a further folding, and then there's the final folding, which is which is going to be take a shape of a globular protein. And because there were two chains. So that's why they will be the quaternary structure. If there was only one chain, then we would only talk about the primary, secondary, and tertiary structure. Then coming to question number 13. Now, which row shows the monomer and the type of bond? Monomer and type of bond that form a polymer. So everybody knows alpha is only in starch and glycogen. So beta is only in cellulose. So there was always some sort of thing which you had to understand. So beta cellulose. So the answer to this was alpha glucose, type of bond 1, 4, and 1, 6, and amylopectin. Then 14, which biological molecules always contain the element nitrogen? Now, DNA, proteins, all contain nitrogen. So, cellulose was wrong. So this is where you have to, lipids was wrong. Here starch was wrong. because that's a carbohydrates. Carbohydrates have carbon, hydrogen, and oxygen. Similarly, fats have carbon, hydrogen, and oxygen. So nitrogen is only present in number one protein. So they gave you enzymes, which was a little bit of so make you realize that enzymes are proteins, and mRNA and tRNA. So all the nucleic acids have nitrogen bases, and that is what you had to understand. Which statement about the quaternary structure of proteins is correct? You see, when the primary structure is made, then of course the folding depends upon the primary structure, and then of course the folding. So which statement about the cotton depends upon the primary structure of the subunits? It has to be two or more polypeptide chains. Now, which set of statements correctly describes hemoglobin? Now, that's, hemoglobin is a globular protein. So the answer to this was A, and the ones which are wrong are the ones which I marked in red. So this was wrong, and this was wrong. So they're not all just helixes. It's a globular, so it would have secondary structure and tertiary structure. Question 16, the diagram shows different types of bonds found in biological molecules. Now, which bonds are found in proteins with a tertiary structure? You know, peptide bond in the primary structure, P for peptide, P for primary, then secondary has hydrogen bonds. Then tertiary structure has DHIV, disulfide hydrogen, ionic, and van der Waals forces or hydrophobic hydrophilic interactions. And then quaternary, of course, is just the putting it all together. Now, if you look at this diagram, which I'm just going to show you, now this diagram shows you the protein and the polypeptide. So you can see this here is the hydrogen bond. Then you can see here this is the disulfide bond. Then the positive NH3 plus and the CO negative, this forms the ionic bond. So this is the ionic bond. Now this is from your book, which is an old edition. And then weak hydrophobic interactions. Now this is between the two non-polar R groups. So you have to know these, you have to know the structure, hydrogen bonds forms where a partial positive and a partial negative on oxygen forms a hydrogen bond, just like water molecules do. And then disulfide is between the two sulfurs. And then NH3 plus and CO negative ionic bond. So between this will be an ionic bond. And then between the two non-polar R groups will be hydrophobic. Non-polar means hydrophobic interactions. So you have to know these to be able to do that question. Now, question 18, the color of a positive Benedict test due to the formation of copper oxide, and the mass of copper oxide is proportional to the mass of the reducing sugar present. So samples of fruit juices were presence of reducing sugar and non-reducing sugars. Using the Benedict's test, the result shows the mass of copper oxide after boiling the Benedicts. So gives you the mass, and after acid hydrolysis and boiling with Benedict's, which sample contains the most non-reducing sugar? So this would be the mass of the reducing sugar, but then which one has more? So this has no difference is 0. This is 30 and 45. So 15 is the non-reducing sugar. Then 50 and 55, 5 is the non-reducing sugar. Then 65 and 75, 10 is the non-reducing sugar. Because you see the copper, the red precipitate can then be weighed. You actually filter it and then you scrape it off the filter paper and you can weigh it. So B was the answer because that had the most non-reducing sugar. So first you do it, so that would be solutions with glucose and sucrose. So the glucose would react with the Benedicts, but then when you took another sample and then you added the acid, so then the glucose would also react and the sucrose would break up into glucose and fructose and would also react. So you would get more mass of the precipitate. Then question 19. Which diagram shows the bond between the individual units of both cellulose and glycogen? So it's basically they're the glycosidic bonds. In one it will be the beta and one will be alpha, but it is of course the glycosidic bond. Which row describes a triglyceride? So question number 20, it's C, hydrophilic. It's not hydrophilic. They can't, fat doesn't dissolve in water, and soluble in alcohol, yes. So that was C. And it says question 21, the diagram shows the amino acid serine. Which circled area is different in all other amino acids? So you know it is different in which circled area is different in all other amino acids. So the answer was C because it's C double bond OH, and this is not the one which was different. And the two is frozen lake walls. After cold winter, mineral nutrients are brought to the surface. Which properties of water contribute to the process? 1 and 3. 1, greater density at 4, and number 3, it's a solvent. Which type of bonds hold together an alpha helix or a beta pleated shade? Now, everybody knows this is the secondary structure, and that is the hydrogen bonds, which describes only the quaternary structure of hemoglobin. So, alpha helix, dipeptide, globular structure, the specific order of amino acid, four polypeptide chains joined together. This was the only possible answer, so that is the answer. The statements are about the properties of water: ability to form hydrogen bonds with other molecules, less dense when frozen, able to hold a lot of heat. What allows a small insect to rest on the surface of the pond? It's only the ability to form hydrogen bonds with other molecules. I think this has been removed from the course, so I don't think we're going to explain it much. Which bonds hold together the structure of cellulose? So basically, cellulose is beta glucose, so glycosidic bond, and then ionic bond between the hydrogen bond between the molecules. So the cellulose molecules then form the hydrogen bonds between them, and this is of course so between the cellulose molecules. So it's 1 and 2, sorry. Then which bonds are found in glycogen? Glycogen, is 1 4 and 1 6. So but it's alpha, it's not beta. So beta would not be, this would be incorrect. So this is the one you've got to keep out of it. So it's 1 4 and 1 6 alpha bonds. Solution of biological molecules are tested for sugar. The table shows the colors of the solutions after testing. 1 is blue, 2 is green, 3 is yellow. Boiled with hydrochloric acid, orange. So this has got sucrose because it was blue previously and now it is orange. So this would be a reducing sugar. So this would have sucrose, which would be a non-reducing sugar, sorry. Green, so that means no reducing sugar, just the reducing sugar. There would be no non-reducing sugar in it. Yellow and red, again, it contains non-reducing sugar. Why? Because initially it was yellow and then it is red after you've added the HCl and done it. So that means it contains glucose and sucrose. Two only contain glucose, one also contains. So which may contain non-reducing sugar, 1 and 3. You see, when you test for Benedict's, then it gives a blue color. But when you test it, the same sample again with HCl and glucose, you get an orange color. So that means there was no glucose in it, but there was sucrose in it. And in 3 it was yellow, so it was a small amount of glucose in it, but then it became red, so it was a large amount of sucrose in it because the sucrose breaks down and breaks up into glucose and fructose. Now, two disaccharides are maltose and sucrose. Maltose is formed from two molecules of glucose. Sucrose is made from fructose and glucose. Fructose and glucose are isomers. Which row shows the molecular formula of the two disaccharides? So C6H12O6 plus C6H12O6 give you C12H24O12. उसमें से minus the H2O because it's a condensation reaction. So it will be left with C11H22O11, and this was the correct answer. Then the diagram shows a carbohydrate molecule. Question 30, the diagram shows a carbohydrate molecule. Now you can see how I have outline the 6 carbon ones, which are all in one direction. If it was cellulose, then it would be, you see the carbon flips. So if you have 1 6 up, then in the next one, the next 6 is down. So you would find it like this. You would find a structure like this. This would definitely be cellulose. So 6, 6, and a 6 down. But here, because it's all 1 4 and 1 6, so it has to be starch and glycogen, and it could not have been cellulose. So cellulose is wrong. 1 4 and 1 6 is starch, and this says amylose. Amylose doesn't have 1 6 bonds, and amylose doesn't have. So amylose everywhere was wrong. Cellulose was wrong. These were the things you had to pick up. If you actually knew this, you could do this. Which of the bonds stabilizing the tertiary structure of protein could be least likely to break when the temperature is increased? You see, the most strongest bonds are the disulfide bonds and the peptide bonds, and these are, of course, the strongest bonds. So this is all a little bit of biochemistry. The diagram shows the structure of 4 amino acids in solution: glycine, lysine, alanine, and aspartate. And which amino acids have no overall charge? And that was alanine and glycine. Well, of course, you've got to know a little bit of the chemistry, so you will easily figure this out. The statements are about the properties of water. It requires a lot of heat to evaporate, retains a lot of heat, able to form hydrogen bonds, and with other polar. Which properties are important in transpiring plants to prevent enzymes in leaves from being denatured? So that was one only, and a lot of heat to evaporate. You see, that cools the plant, so then the enzymes will not be denatured. 34, which shows the basic unit of glycogen. That's alpha glucose, and you have to look at the chemistry of all of them. And I always tell you to remember it: alpha below, beta above, and the mnemonic becomes A, B, V, A. Whether you want to call it the musical group ABBA or you want to call it ABBA. And that's on carbon 1. So alpha below, beta above. So alpha below here, alpha below here, and the rest are, of course. So this was wrong, and of course this one is the one which is the critically wrong one. So B was correct, and this is how you had to figure it out. Then 4 solutions, sugar solutions were tested. Benedict, the table shows the color of the solutions after testing. 1 was green, 2 was blue, 3 was brick red, 4 was yellow. And which is the best interpretation of the results? Now, this, of course, is a very, very tricky question. I usually find them very interesting because you see they gives you all this. And so 0.05 reducing sugar, zero point 0.5 would be more than 0.05 non-reducing sugar, then 1% reducing sugar, and then 0.1 reducing sugar. So 0.1 is the one which is the least, and then, no, sorry, 0.05 is the least. So 0.05, 0.1, and zero point 0.5, and then 1. So this is how you would arrange it, and it would be, of course, A, the answer would be A, but this is what you have to pick up. What was the least amount? So 0.05 least, this one, then 0.1, then 0.5, and then, so it should have been 1423, 1423. So this is how you'd have to figure it out, 1423. So 1, 4, 2, 3. 6. Which features adapt a cellulose molecule for its function? Long chains of glucose coil into helix, rubbish, many hydrogen bonds, and insoluble. So that was 2 and 3. The structure of phospholipids and triglycerides include the following: glycerol linked to fatty acids, glycerol linked to fatty acids, hydrophobic fatty acid chain, saturated fatty acid. Which structure enables the formation of a lipid bilayer in cell surface membrane? So this is the question. The lipid bilayer is because the tails are oriented in towards the center. So this is the part which is hydrophobic. So that is why the answer was the hydrophobic fatty acid chain. They say which enables the formation of a lipid bilayer. And that was the only answer to that. Then coming to question number 38, which molecules contain a glycosidic bond? Now there are of course 4 of them here. I will have to just slide it a little up. And so you have to understand glycosidic bond, and that was, of course, you have to pick. It's a little difficult. I find it a little difficult for you all to do this. So this was the answer was C. 40, which shows beta glucose. Now you've got some beta is above, so the above are only A and C. These were all below, so this was wrong, and then you have to figure out between A and C, and then of course this is the wrong part which I have circled. So I want you all to understand why this was wrong. You see, in MCQs you've got to understand, you have to really explain to yourself why which one was wrong. Why was B, C, and D wrong? If you don't do that, well, I'm sorry, then you can't really be doing A-level biology. We finished 40 questions on this, and this will go on to, so this is the video 1, and there is another video 2 which will be another 40 questions which we will handle in the video 2. So please everybody look, I'm going to post it tonight, and so you have that to revise your biological molecules chapter. And please do get back to me if anybody needs the a PDF file of this MCQ, then you can WhatsApp me on my phone number and I'll send it to you. And thank you very much.